Over 30 million people have spit into a plastic tube and mailed their saliva off to a commercial DNA testing company. What comes back a few weeks later is usually a neat little pie chart, dividing a person's identity into strict percentages and hard geographic borders. But geneticists point out a major flaw in how this is calculated. These companies rely heavily on their own private databases, using modern proxy populations to guess where your ancestors lived centuries ago. The algorithms run on a model called discrete demes. They take a messy, interconnected human history and mathematically force it into isolated, artificial boxes based on today's geopolitical borders. In reality, human genetic variation ignores those borders. It is a continuous, overlapping gradient. These proprietary charts are just one interpretation of the code. Geneticists are increasingly looking past the percentages to find the specific anomalies that these maps often miss. To track true ancestry, you have to look for the evolutionary glitches. When early humans shifted toward agriculture, our diets changed, and our genes had to break and rebuild themselves to keep up. Take the AM1 gene. In early farming societies, humans developed extra copies of this gene, flooding their mouths with an enzyme to break down dense starches before the food even hit their stomachs. Then there is the LCT gene. Every human is born with the ability to digest milk as an infant, but the original factory setting of our biology turns that ability off in childhood. Some groups developed a mutation that left this switch permanently stuck in the on position, allowing adults to digest dairy. This is a textbook case of convergent evolution. The exact same dairy-digesting mutation appeared independently across 3 completely disconnected populations in Scandinavia, East Africa, and the Middle East. The environment forced the issue. To survive in harsh, arid landscapes, these early humans had to rely on cattle herding, meaning those who could extract calories from milk lived to pass on their genes. Your ability to eat a bowl of ice cream today is a biological receipt of your ancestors' highly specific agricultural survival tactics. Beyond dietary shifts, moving into extreme climates forced the body to develop physical defenses against the landscape itself. When faced with deadly stomach bugs, some populations developed a broken version of the FUT2 gene. This mutation acts like a cellular bouncer, stripping away the specific sugar coatings on the intestinal wall that the norovirus needs to latch onto. Because stomach pathogens were a global threat, this exact genetic defense evolved independently in populations with European, Bangladeshi, and sub-Saharan African roots. In the Arctic, survival meant fighting off freezing temperatures. Populations in Siberia and Northern Europe developed a hyperactive version of the UCP1 gene, turning their brown fat cells into an automatic internal furnace that burns energy as heat. Meanwhile, East Asian ancestors faced extreme heat and humidity. Their bodies responded with a mutation to the EDA gene, drastically multiplying their sweat glands to dump body heat at a much faster rate. Human expansion out of Africa was made possible by localized, desperate genetic breaks against climate and disease. Evolution never provides a clean upgrade. It is a messy series of biological compromises with lasting consequences. The TRPM8 gene gave Northern Europeans a highly sensitive cold warning system to keep them alive in the winter. But that heightened nervous system response accidentally caused a lifelong susceptibility to severe migraines. For Inuit ancestors living on a diet composed almost entirely of seal and salmon, the body completely deactivated the FADS gene. Because they were eating so much preformed fat, they abandoned their internal fat conversion processes entirely. When people with that specific deactivated gene consume a modern diet heavy in plant-based seed oils, their bodies cannot process the excess omega-6 fats, triggering severe, system-wide inflammation. When our environments change faster than our DNA, ancient biological victories transform into modern physiological liabilities. By studying these ancient, localized genetic glitches, researchers are reverse engineering modern medical cures. In a small population in western Finland, scientists discovered a mutation in the SLC30A8 gene. This glitch essentially broke the body's standard regulatory brake pedal that stops insulin release when blood sugar spikes. This chart illustrates the result of that broken gene. Compared to the general population's high baseline, the type 2 diabetes risk for this isolated Finnish group drops 65. Pharmaceuticals are now attempting to artificially replicate this exact sequence to manufacture a global diabetes cure. Studying isolated population genetics is about mining ancient biology to cure the future. Look past the percentages and the national borders on your commercial results. Every human genome presents an overlapping mosaic of desperate, brilliant survival events. The human genome was fundamentally forged through relentless, brutal exposure to freezing ice ages, widespread famine, and deadly ancient diseases. Your ancestry is the living, biological record of exactly how your family survived.